All right, so my name is Sarah Dean. I'm an assay development scientist here at Tricor, and today I'm going to tell you the story of how the mammal got its placenta. Because in a past life, I studied microbiomes, and I love to speak up for our misunderstood microbial friends as often as I can. I did give a version of this talk previously at TriTalks, and I noticed that some people in the audience were there. So I get twice as much time, so I get to add a little more detail this time. So I'm glad to be able to present it again. Um, so when I was a kid, I loved Just So stories by Rudyard Kipling. You know, there's the stories of how the elephant got its trunk, how the camel got his hump, how the leopard got its spots. And of course, those are works of fiction, but the Just So story I'm going to tell you today is true. The placenta, of course, is the organ that allows us to grow our babies inside of us and give birth to live young. Long pregnancies are important to humans because they allow our big brains time to safely grow inside of our mother before we are pushed out into the world. Long pregnancies are understood to support human intelligence. But this is not just a story about mammals such as humans, it's also a story about viruses. Right, so you probably think of yourself as a human, but you are actually a human microbe hybrid. One way you're a hybrid is due to your microbiome, which gets a lot of press these days. Maybe you've already heard that you carry 10 times more bacterial cells than human cells in and on your body, resulting in 100 times more microbial genes than human genes being expressed in and on your body. These microbes and their genes carry out essential functions for you. They produce essential vitamins, they help you digest your food, they train your immune system, and they even wield influence on your mood and your behavior by messing with your hormones and neurotransmitters. But there is another much deeper way that you are a human microbe hybrid, one that doesn't get a lot of press, maybe because it's a little uncomfortable to think about. Your DNA, your genome, is part human and part virus. And some of this viral DNA gives you key features that are at the core of who you are. You see, there's this group of viruses called retroviruses that genetically modify their hosts. Thanks to them, about 8% of your genome is made of viral DNA. So today I'm going to introduce you to a virus that has become a part of you and that is responsible for the placenta that grew you. But first things first, before we can understand how retroviruses gave us mammals our placenta, we have to answer the question, what is a virus anyway? We're all familiar with the computer virus, which is a bit of computer code that hijacks the function of a victim's computer. These were named viruses for a reason because this is exactly what real viruses do. Well, not in computers, of course, but in a living cell. A virus injects genetic code into a host cell, and that genetic code hijacks the cell. The cell is now under the control of the virus, and its main purpose in life becomes to replicate the virus. Anatomically, viruses are just genes in a box. This means they are rogue genetic material not under the control of any living organism. They are far smaller than a cell and much too tiny to carry with them a complete toolkit for life. They don't have any of the necessary equipment to harvest energy from their environment, to replicate, or even to use any of the instructions inside their genome. So they infect cells so that they can use the cell's metabolic equipment, reproductive equipment, and gene expression equipment. So viruses only have lifelike qualities while they are infecting a cell. Because of this, they do not quite make the cut for the title alive, but they do control their own transfer from victim to victim, and they display a number of lifelike qualities once they are inside a cell. So viruses certainly occupy a gray area between the living and non-living worlds. So next, a quick and dirty introduction to retroviruses specifically. Retroviruses have genes which include all the instructions they need to infect cells, genetically modify them, and build new viruses. I said viruses are genes in a box, and this box is called a capsid and is made out of a protein crystal that safely transports the viral genetic code from one victim to another. All viruses have this, and yes, it is a crystal, a fact I have used to sell my daughter on viruses when she was 5 years old, because anything that's a crystal is beautiful, of course. Crystals are made when identical molecules or elements come together in repetitive patterns. The small protein pieces of the viral capsid are identical to each other, and they adhere to each other naturally due to their chemistry, coming together around the genetic material in a repetitive pattern. Retroviruses are also enveloped by lipids, which are like fats. Fats are a type of lipid. They use these to fuse with the cells that they infect. The lipid envelope is just like the membrane of a cell because a virus actually steals it from the cell that birthed it. On its way out of its cell of origin, it cloaks itself in some of the cell's lipid membrane. So retroviruses are rogue genetic elements inside a crystal protein capsid inside a stolen lipid envelope. Retroviruses also have fusion proteins, sometimes called spikes, which you have, of course, heard about in the news in relation to SARS-CoV-2. SARS-CoV-2 is not a retrovirus, but many viruses share a similar anatomy. They are called fusion proteins because they also help the retrovirus fuse with the cells that they infect. Without fusion proteins, the virus would be completely harmless, unable to enter a victim. So here, the flat gray line represents the cell membrane on the surface of a cell, and the gray line around the virus represents its lipid envelope. The fusion proteins in orange bind to molecules on the cell surface in blue, allowing fusion between the viral envelope and the cell membrane, like the fusion of two soap bubbles into one larger bubble. And I promise this is very important to the placenta story. All right, so in this image, here is a retrovirus and here is a cell. Maybe it's even one of our own cells, one of the trillions of cells that make up our bodies. This inner circle is our cell's nucleus, which shelters our DNA library, our genetic code. So retroviruses fuse with their victims to infect them. Here, the viral lipid envelope is studded with fusion proteins in red, which bind with some structure on the cell surface, allowing the viral envelope to fuse with the cell membrane, emptying the viral capsid into the cell. Our cell is now infected. The virus uncoats, releasing viral genes into the cell, which it inserts into our genome, genetically modifying us. Once it is part of our genome, we call it a provirus. At this point, the provirus may simply lie latent in our genome. When this cell replicates, its daughters will inherit it, just as they inherit every other gene in the parent cell's genome. The virus is now a permanent part of this cell's lineage, passing from one generation to the next. If this is a cell that produces sperm or egg, any baby that comes of it will also inherit the provirus. Our cell could also begin expressing the provirus's genetic instructions, meaning our cell unbewittingly builds new retroviruses from the provirus DNA, producing new viral genomes, new viral fusion proteins, and new viral capsid proteins. The pieces of the viral capsid crystallize around the viral genomes and bud from our cell, taking its fusion proteins and a piece of our cell's lipid membrane with it. All the baby virions float away to infect neighboring cells. So proviruses have a latent and an expressed phase. In the latent phase, the retrovirus passively replicates when the cell divides, and it is inherited by all descendants of the infected cell. So sometimes latent proviruses carry genes that cause the host cell to proliferate more rapidly, resulting in more copies of the cell and therefore more copies of the provirus that the cell contains. The consequence of this can be cancer. So a tumor is what you get when your cells keep dividing and proliferating when they shouldn't, and many retroviruses are linked to cancer. In the expressed phase, viral genes are expressed by the host, producing new viruses that are released to infect new cells. Whether expressed or latent, a virus will need to evade its victim's immune system to persist inside the host for any useful length of time. Many viruses carry genes that help them suppress their host's immune response, giving them time to reproduce. Retroviruses need a special set of tools to infect and genetically modify their victims, and their genome includes the instructions for these tools. We know what these instructions look like, so we know retroviral genetic code when we see it, even when it's hiding inside our massive human genome. And this allows us to find retroviruses using DNA sequencing. Retroviruses can be very damaging. So an example of an infamous modern retrovirus is HIV. You can never get rid of this virus once you have it because it becomes part of your human DNA. We call viruses like HIV exogenous retroviruses because they came from outside of us. We were infected from an outside source. Exo refers to outside, like exit. But retroviruses have been around infecting our ancestors for a long time, and they've accumulated in our genomes over the eons. Most of these ancient retroviruses are now silenced and defunct, but we can still identify them by those genetic sequences that are characteristic of all retroviruses. These viruses are called endogenous retroviruses. Endo refers to within, because they are now part of our genome and they come from our own ancestors, not some outside source. But some endogenous retroviruses are not defunct. Some have provided us with novel genes that have evolved us. Domesticated is the term we use for retroviruses whose genes we actively use. And I used this image because the shadow there reminds us of the domestic dog's past as a dangerous wolf. Our domesticated retroviruses may have a dangerous past, but we've captured them and we've made them our own. Right, so now that we've had a mini retrovirus lesson, we can get to the meat of the story. One incredibly interesting example of a domesticated retrovirus is the one that gave us mammals our placenta. Mammals are characterized by growing hair for warmth, producing milk from mammary glands. We mammals are named for our mammary glands, and giving live birth. So instead of our eggs maturing outside the mother, mammalian eggs mature inside the mother. Our fetuses are nourished by a placenta instead of by the egg yolk and egg white. The placenta is interesting because it's an organ that's built out of both maternal and fetal tissues, which allows the mother to nourish the baby and remove its wastes. So in this image of the placenta, everything on the red spectrum is maternal tissue. The blue line is the outermost portion of fetal tissue known as the chorion. It is made out of fetal cells, and it represents the boundary between the maternal and fetal portions of the placenta. The chorion is what invades the mother's uterus to nourish the fetus, and it forms a protective barrier, the blood-placental barrier. Bird and reptile eggs also have a chorion, which is also the outermost layer of fetal tissue. Homologous to the mammalian chorion, it provides protection to the growing baby and is involved in providing the embryo with nutrients and oxygen and removing wastes. In mammals, the chorion grows into these wavy structures, which we call the chorionic villi, which behave quite strangely. So, for example, the cells of the mammalian chorion proliferate very rapidly, aggressively forcing their way into the mother's uterine lining, bursting her blood vessels left and right so that her blood washes over the fetal cells, transferring all their nutrients and oxygen to the baby. And this is very virusy behavior. Remember, retroviruses commonly cause excessive cell proliferation. Typically, our immune systems wouldn't tolerate this type of parasitism and dysregulation, but the fetal portion of the placenta also dampens the mother's immune response, another behavior typical of viruses. But perhaps the most virus-like feature of the mammalian chorionic villi is fusion between cells, facilitated by fusion proteins. So remember this image, which describes how enveloped viruses enter cells? Viral fusion proteins in orange allow fusion between the viral lipid envelope and the lipid bilayer of the cells that they infect. This layer of blue cells are the chorion produced by the growing mammalian baby, and they all fuse together, becoming like a large single cell with multiple nuclei. The reason these cells can fuse is because they express a retroviral fusion protein called syncytin. How do we know it's a retroviral protein? Because it's found smack in the middle of stereotypical retroviral code. Placental syncytin is a viral fusion protein expressed by animal cells. And this fusion is absolutely essential to placental function, because we have these immune cells that squeeze between the other tightly packed cells of our bodies in order to reach sites of infection. If they could, a mother's immune cells would squeeze across the placental barrier and eat up the parasitic fetus. Happily, there is no between cells here, since what we have effectively is a single continuous cell, and this protects the mammalian fetus from attack by the mother. And actually, syncytin is not the only domesticated retroviral protein important to the placenta and gestation. The placenta's genetic code is actually rife with domesticated retroviruses, which increase the rate of cell proliferation, attack maternal cells, and release chemicals that dampen her immune system. Mammalian fetuses infect their mothers because they express retroviral genes. There have actually been experiments in which researchers modify laboratory animals to block and silence those retroviral genes. And guess what happens? They lose their pregnancies. Without retroviral activity, the placentas don't attach properly to the uterine lining of the mother. They don't invade maternal tissue, so they fail to supply nutrition to the fetus, and the fetus ends up being attacked by the mother's immune system. This shows us how important retroviruses are to successful placenta formation. Another way we know how important retroviruses are to placenta formation is by looking at mammals that don't have placentas, like platypuses. Platypuses are mammals because they have hair and they produce milk, but they are remnants of an ancient mammalian group called monotremes that predated live birth, who still laid eggs like their reptilian ancestors. While monotremes were once quite common, only 5 species have survived the ages and are still around today. And the genetic code for the platypus chorion does not contain retroviruses like that of placental mammals. So here's the story. Once upon a time, one of our ancient egg-laying mammalian ancestors was genetically modified by a retrovirus, giving it special mutant powers. The retroviral genes enabled fetal tissue to invade the mother and fuse to produce a solid continuous barrier that blocked her immune response to the invasion. Domesticated retroviruses allow mammalian babies to infect and parasitize the mother for extended periods of time inside the womb, a relationship we call pregnancy. Now, maybe you're skeptical because you might know that some non-mammals also give live birth, like some fish and reptiles. It's hard enough to believe retroviruses created placentas even once. It can't possibly have happened a whole bunch of times, right? Well, the Mabuya lizard is a skink with a very invasive and mammal-like placenta. And guess what? We find retroviral DNA in its genome that control critical aspects of its placenta formation. And other skinks who lay eggs, like normal lizards, do not contain these retroviruses. The Mabuya lizard retroviruses are different species from the ones in our genome, but they function in the same way, using viral proteins to invade maternal tissue, create a barrier against her immune system, and disarm her immune system. A group of spiny rayed fish known as percomorphs are known for their tendency to give birth to live young. This group of fish also have a retroviral fusion protein encoded in their DNA. This is an ancient group of fish, and this gene is the oldest known retroviral gene in vertebrates, estimated to have been inserted into the percomorph genome 110 million years ago, resulting in ancient fish placentas. Placentation, or the development of a placenta, has evolved separately many times in the animal kingdom, and everywhere we've looked so far, we've found retroviruses responsible. There's still many other placentas that we haven't checked for retroviruses yet. Many sharks also give live birth, as well as a number of snakes, such as anacondas and vipers. But given the emerging pattern, if I had to place a bet, I'd bet retroviruses are responsible for these placentas too. So if retroviral infection can evolve placentas in so many animals, can they also evolve other animal features? I mean, do retroviruses only make placentas, or do they have any other talents? So there's compelling evidence that retroviruses genetically modified the brains of us land vertebrates, increasing the efficiency of information transfer between neurons and enhancing our brain plasticity, which is another word for adaptability. So this means retroviruses have made our brains more efficient and more adaptable. They also seem to help protect our brains against the toxic effects of other retroviruses like HIV. So if you're interested, you can check out research on ARK and HERVK. These are a couple of interesting retroviruses in the land vertebrate brain. It may not just be in the brain that our domestic retroviruses protect us from exogenous retroviruses. In human cells grown in petri dishes, researchers have watched human domesticated retroviruses produce a variety of products that interfere with HIV, preventing HIV from infecting human cells and from replicating. Perhaps in some primitive way, our domesticated retroviruses are defending their territory, like a guard dog. Time will tell if our retroviruses work to fight HIV in our bodies as well as in the petri dish. Telomerase is a complex of genetic material and protein that builds telomeres on the ends of our chromosomes. Telomeres act as aglets for our DNA, protecting the ends from damage, and it too is of retroviral origin. We also know that certain hormone glands express a lot of products from domesticated retroviruses, but right now we don't know what they do for us. So a number of different tissue types appear to actively use their symbiotic retroviruses day to day. Still, researchers do wonder why the placenta seems to be the main target for retroviral modification, especially the placentas of higher mammals, which are shaped by a cornucopia of retroviruses, far more than any other tissue type. The placenta is the most diverse organ across the animal kingdom because different animal groups have been invaded by different sets of retroviruses. Each time a retrovirus invades, it donates its unique fusion proteins, immune suppression proteins, proliferation genes, and whatever else. The reason placentas are so diverse is that they are rapidly evolving, changing every time a new retrovirus inserts itself into some species' genetic instructions for the chorion. The placenta seems to be a site of rapid retroviral innovation where these viruses throw all their proverbial spaghetti at the wall to see what sticks. And we don't really know why yet, but here is the current prevailing hypothesis. So remember, in reptile, bird, and monitoring mammal eggs, the chorion is the outermost layer of fetal tissue. And in the placenta, it is also the outermost layer of fetal tissue. Whether hatching from an egg or birthed from the womb, the chorion is discarded after birth. The chick will leave it behind with the broken egg, and the placenta is birthed after the baby and either eaten by the mother, thrown in the garbage, or left to rot somewhere in the natural landscape. Now, in most cells of the body, retroviral DNA is silenced. How? By adding little methyl molecules to the viral DNA that turns it off. Why? Because retroviruses introduce change, and change can be dangerous. Retroviruses disrupt genes that were functioning perfectly well. They change how our DNA is expressed. They wreak havoc to the law and order of our bodies. So we turn them off. But it appears this retrovirus silencing does not happen in the chorion, allowing the chorion to express any retroviral fusion and immune suppression proteins it might have. This has resulted in the chorion producing placentas on repeated occasions by invading maternal tissue to some extent or another, fusing and blocking her immune response. The reason the fetus can afford to not turn off retroviruses in the chorion is probably because the chorion just gets thrown out after birth anyway. Most of the problems that could be caused by retroviruses won't affect the baby because ultimately the chorion separates from the baby. So embryos and fetuses allow those retroviruses to speak in the chorion, which means the chorion benefits from all that retroviral innovation while experiencing minimal risk since it is disposable. Retroviruses probably don't target the chorion more than they target other tissues. It's more likely that the chorion just doesn't silence its retroviruses the way other tissues do. So all the bizarre retroviral activity is on display there. So I hope I've convinced you that retroviruses have given us important defining features. For example, we usually assert it is our intelligence that sets us apart from the rest of the animal kingdom. Extended gestation allows our large brain time to develop in the safety of our mother's womb, a benefit provided to us by retroviruses. And brain efficiency and plasticity have been greatly enhanced by retroviruses, setting the stage for the high-powered human brain to evolve. Additionally, we know there is a slew of other retroviruses whose role in the human body we have yet to define. Retroviruses are more than just part of our human microbiome. They're part of our genome. We are literally human virus hybrids.